Hello, friends. Welcome to episode 9 of Genomics for All. So, December 1st is World AIDS Day, and apparently, we have to talk about awareness for AIDS, HIV, and most importantly for us, genomics for AIDS. What genomics holds promise for the diagnostics, for the treatment, etc., for AIDS. Now, before going into the details, what is AIDS? For people who are totally unaware, AIDS, as the abbreviation suggests, it's acquired immunodeficiency syndrome. And as the name suggests, it's an immunodeficiency syndrome where the immune system gets compromised. And this is caused by a virus whose name is also very apparent, human immunodeficiency virus. Now, human immunodeficiency virus is the virus that is the causative agent for causing acquired immunodeficiency syndrome. Now, this is the virus, this is the disease. Now, what is this virus all about? It is a retrovirus, which is a virus having two strands of RNA. And this is a very peculiar virus in itself because it's an RNA virus, but it makes DNA from this RNA, which is defying the central dogma of life, wherein RNA is made from DNA. We talked in the previous episode that the central dogma of life is RNA made from DNA. But this virus is a very peculiar virus. It makes DNA from RNA, that is the genetic material of this virus, and the DNA gets integrated into the host genome of blood cells, that is the T cells in our body, and that is how the whole it attacks the immune system. So, how does it do it? This virus uses a specialized enzyme called reverse transcriptase and a specialized process called reverse transcription, as the name suggests, RNA to DNA. It can be dormant for years into the genome of the human, and there are various mechanisms suggested when it gets activated and the whole process of viral propagation takes place in the cell. The whole immune system gets compromised, people die. There are therapies, retroviral, antiretroviral therapies which work, but then the whole disease, AIDS and HIV, is still a challenge for physicians and scientists all over the world. What can genomics do? What can we do at MyBiome Therapeutics? We always talk about what can next generation sequencing and genomics do for AIDS. Now, there are two aspects. One is the viral genomics, wherein, as I said, it's a retrovirus. So it has about 9 different genes coding for 16 different proteins, 10 kilobase per virus. It's a very small virus, like the COVID virus. Very small virus, but can cause damage to as big as death. How can viral genomics be useful? Is the first thing is the surveillance, wherein viral genomes can be queried, data mined for surveillance. Either it could be drug resistance or it could be any therapy. So researchers all over the world and clinicians all over the world are looking at different genomes, different polymorphisms that occur in this virus. As you all have heard about polymorphisms, etc. in COVID, similar things happened in AIDS virus for a long time. So those people are still understanding those polymorphisms or changes in the virus and understanding more about the drug resistance and other surveillance mechanisms. So that's some about the viral genomics part. Now, we humans also have a genome, and it is getting integrated into the human genome. So does it mean that it is uniform across all humans? Now, this is very interesting that there has been reports suggesting that different humans will have different predisposition to HIV. Now, there is no very concrete scientific evidence, but whatever evidence exists is very exciting. Now, can we map human genome and say whether a person will be more susceptible to AIDS disease? These are possibilities, and these are done by a study called Genome-Wide Association Study, GWAS, wherein many human genomes are sequenced and then looked whether some humans are more susceptible, different ethnicity groups, different race groups, etc. From different continents. So very exciting studies are being conducted to see the differential susceptibility of humans to this AIDS virus. And lastly, I wouldn't go too much deep. There's a lot to talk about AIDS, HIV, genomics for AIDS, but I would end with some hopes and some hypes. So now, what are these hopes? We have this virus integrated into the genome. Now, a very exciting scientific study suggests that you can use CRISPR which are the molecular scissors which can precisely cleave out a genomic DNA of our choice. Now we can use CRISPR-Cas9, this technology, molecular scissor technology, to actually cleave out the HIV that has been integrated into the human genome. Now this looks like science fiction, but it is not. This is a reality wherein you can actually cleave out the HIV virus that has been integrated into the human genome by using molecular scissors like CRISPR-Cas9. With hopes come hypes. Now, you all would have seen the famous twin Chinese babies that was created, and these babies were defined to be HIV free. How did they do it? The idea was very simple. There is a receptor on the blood cells called CCR5. These are chemokine receptors. Now, viruses use this receptor to internalize and then propagate inside the cell and damage the immune system. So, the idea the scientists came up with was very interesting, that you delete CCR5, the receptor gene, and now you do not have the receptor, therefore, the virus cannot internalize. And those babies were claimed to be HIV free. Then came the whole ethical considerations. That's why I'm calling it hype. And that project was abandoned. Time is not yet right to have all this right now. So we don't discuss that after that. But that is one thing I wanted to bring up, that this is possible. This is not science fiction. And there are technologies to do this, but in the we have to do it in the framework that is defined by the government, etc. So I would end with this, and I would request all of you to raise more and more awareness about HIV and AIDS this month, especially when it is the World AIDS Day on December 1st. Thank you so much.